Hello everybody, it's me Antonio, and for today you're going to learn the animal mitosis using a microslide viewer by the National Teaching Aids Incorporated. So the following slides would show you a magnification of 750 times. This means that the microscope was set at that power when the photograph was taken. So now let's begin. This slide shows the zygote, the fertilized egg of the Ascaris. Two masses of chromatin can be seen in the cell. One mass was the nucleus of the egg. The other came from the sperm that fertilized the egg. The sperm and the egg are very different in size and shape, but the chromatin contributed by each is equal in amount. Each parent, therefore, supplies an equal amount of hereditary material to the offspring. The chromosomes are not seen distinctly because they are very thin. This cell is thick and round like a tiny ball, making it difficult to keep all the chromatin in focus. The next slide shows you the Metaphase. The chromosomes have become much shorter and thicker so that they are now easily seen. Each parent supplied 2 chromosomes to form the zygote. Notice how the chromosomes begin to pair up. Study their shapes carefully. One pair looks like 2 horseshoes. The other pair resembles bent rods. If you move ahead to slide 4, you can get a better idea of the peculiar shapes they have assumed. In these slides, we shall refer to these chromosomes as the U-shaped and the bent. The sperm supplied 1 U-shaped and 1 bent chromosome. The other U-shaped and bent chromosomes came from the egg cell. After fertilization, the 2 U-shaped ones pair up, as do the 2 bent chromosomes. After a while, the 2 pairs begin to move towards the equatorial plate. At this stage, or slide, this is now the metaphase. In this slide, the chromosomes have moved to the equatorial plate. The equatorial plate is the imaginary line running from E to E. At each pole is a centriole, which is labeled P. A star-like structure called an aster radiates from each centriole. Fine spindle fibers go from the centriole at P to each chromosome. Some of these can be seen in this slide. At metaphase, chromosomes in most cells can be seen to be divided lengthwise, but the parts are held together by a structure called a centromere. Now let's go to the next slide. So this stage is still the metaphase, but showing you a polar view. This is the same stage in the development of the egg as shown in slide 3, but we are now looking at the cell from the side. That is, from one pole through the equatorial plate to the other pole. The 4 chromosomes are seen as they lie flat on the equatorial plate. Look back at slide 3. You can see the chromosomes from the side. The centromeres, which are not visible, would be near the center of each chromosome. Now let's go to slide 5. Slide 5 shows you the early Anaphase. Each chromosome has completed duplication. There are now 4 U's and 4 bents. In these 8 chromosomes, there is enough hereditary material for 2 cells. At this stage, You can also see that the duplicated chromosomes are beginning to form two groups. The groups are starting to move towards the poles, and as they progress, the separation between them will be clearly seen. Each chromosome is being pulled by a spindle fiber attached to each centromere. Now let's go to the next slide. In this stage, you would see the anaphase. The 8 chromosomes are now separated in two groups, each with 4 chromosomes. The spindle fibers are pulling each group of chromosomes towards the aster and centriole at each pole. Electron microscopes show that the spindle fibers are made up of extremely small structures called microtubules. Look closely at the chromosomes. Some of them appear to be beaded in places. Authorities on this subject believe that the long, thin chromosomes become short and thick by twisting into a tight coil. Sometimes the coil loosens up a bit, making the chromosome look beaded. Now let's go to the 7th slide. In this slide, you would see the telophase. The two groups of chromosomes have drawn completely apart from each other. At the same time, the rest of the cell is beginning to divide. The cell membrane pinches inward and the cytoplasm is dividing into two masses. If you go back to slide 6 and examine the cell membrane carefully, you will actually see the beginning of this pinching process. As the cell approaches its division into two cells, the individual chromosomes become less distinct. And now let's go to the last slide. This is now the late telophase. The separation is now completed. The original zygote of the Ascaris has divided to form two daughter cells. Each of these two cells has 4 chromosomes. The chromosomes in each cell will soon lose their clarity. They will become long and thin and not so distinct. Then the process of mitosis will start again in each of the 2 daughter cells. The result will be 4 cells, and these in turn will start the process of cell division of mitosis again. You can thus see how mitosis in these slides achieve its function. One cell with 2 pairs of chromosomes form 2 cells, each a duplicate of the original. In the human being, the process of mitosis is the same, but the number of chromosomes is 46 as compared with the 4 in these slides. There you have it. You've learned animal mitosis using a microslide viewer by the National Teaching Aids Incorporated. Thank you very much for watching and listening. Have a good day. Bye.